Okay, so you will see it will be very different. Almost no equation, and anyway, I want to go to the blackboard, but you will see a lot of membrane that at least Marcus introduced very nicely this morning. But I will show you how we just deal with them experimentally, and yeah. So I wanted to introduce myself first. So for those who don't know where the Curie Institute is, this is a map of Paris. This is a river. This is somewhere we have Notre Dame, which is rebuilding now, and the Pantheon where Marie Curie is buried, and our lab is here. And this is a different part of the Curie Institute, as you can see here. So we are in the middle of Paris. Okay, that said, I will start with some images, at least movies of cells, because I think we heard about different, I mean, we saw nice condensate this morning in cells, but basically there was to stress that we can do a lot of biology without membranes. And I would say membranes are very, very important. And I will show you here 4 examples that I'm going to describe to detail now. That's an example of cells. But here we have a cell. So you can see the cell is migrating, is moving on the substrate. And you can see that cells, at least the front edge is not smooth, it's deformed when the cell is migrating. You can see that the cell behind is leaving a lot of long tetters. Yeah, as you can see here. That's here another example where you can see that the cell is able also to change topology. When the cell is dividing, you start from one cell and you end up with 2 cells. And you can see that the shape of the cell changed a lot during that time, because you see the cell is rounding at some point, then is extending. You can see there is a tiny bridge which is formed between the 2 cells, and eventually that cells divide. So you start from 1, you end up with 2. So in terms of topology, meaning you change topology. This is also another example where the cell is changing topology, but from a different way. So what you have here, this is a cell which is lying on the surface, and this cell is intoxicated by a toxin. And that toxin is used by the bacteria, which is, in this case is Staphylococcus. And this Staphylococcus opens some big tunnel through the cell to go through the blood vessels and to infect after that the organism. So what you can see that the cell originally is like you can imagine is like a surface. And you see the formation of this big hole into the cell, which eventually close back. So again, showing that the envelope of the cell itself is changing a lot of shape and topology. And the last example here, that's a different scale. So now we are looking at what is going inside the cell. So the cell here has some organelles which are labeled fluorescently. And you can see that here, this is what is called the Golgi apparatus. And you can see that from the Golgi apparatus, you have many small vesicles. So this one, they are surrounded by a membrane. And you can see many vesicles which are formed from the Golgi here, and that traffic inside the cells. And the typical size of this vesicle is of the order of 100 nanometer. So let's say that to explain that, to do all this different transformation, so the cells need to change shape. And so it means that there are many processes which are in place, which are able to change the shape of its envelope, not only the interior, but the shape of the envelope. And this requires proteins and a cytoskeleton, generally speaking. So if you have any question, feel free to stop me. Huh? Just big signs. Sure, tell me. So what you can see here, this is the formation of this vesicle, and this scheme here, this scheme here is to show that the cell is using the formation of these small vesicles to communicate between organelles. So the cell is, we have seen this morning, the organelles which are not surrounded by a membrane, but there are many of them which are surrounded by the membrane. First, the nucleus here. But this apparatus, which is called the Golgi apparatus, which is kind of the sorting element of the cells, where proteins are usually produced integrated in membrane here at this compartment, which is the endoplasmic reticulum, and they are transformed here at the Golgi. And to be transported from here to here, yeah, there are this small vesicle that you could see before, and you can maybe see here, no? No, doesn't run, but it's, anyway, the one you have seen before, which are used to transport protein which are produced here at the ER level to the Golgi, and so on. So you have also protein which are transformed at the Golgi that are selected in different vesicle and transported to the plasma membrane, and so on. So meaning that to transport this protein or this lipid through the cell, you have to form this tiny vesicle or tiny tubes. So this suggests that you need for forming this vesicle to deform the membrane to a large extent, right? And this suggests they require different steps. So if you want to form a vesicle, you need to, so imagine that you want to transport a protein from one compartment to another compartment. So you are going to form a vesicle, especially if it's a protein which is not soluble. So what you do is you have a protein here which will be locally enriched. So you will get few copies of protein which are locally recruited. And you need a different process which are able to transform this flat membrane into a small vesicles. And I will explain later how it goes, right? But you need to deform the membrane to somehow form a structure which will be a small vesicle. And to do that, at some point, you have to have deformed the membrane into a spherical object or a tubular object, and you have to cut it because you have to detach it from the donor membrane. And then you have to transport it to another membrane with different transport systems, which are called molecular motors. And eventually, you have to fuse this object with an acceptor compartment. It could be, for instance, the envelope of the cell, which is called the plasma membrane. And then eventually, after fusion, you will have delivered your protein to the acceptor compartment. So this requires different steps where the membrane is first deformed and cut, and after that fused, right? So that's the kind of a thing I'm going to discuss today. I mean, in more detail, how it's done from the biological point of view and from physical point of view. So if I want to be a little bit more precise about a cell, so what we have here is a cartoon where you see all the compartments which are surrounded by a membrane. We don't see the condensate from Will this morning, but we see that we have here the envelope of the cell, which is called the plasma membrane. Here we have the nucleus, and you can see here again the endoplasmic reticulum. We can see the Golgi, and we can see many small organelles which are called endosome, lysosome here. And this structure here are called mitochondria. And this is the place where the energy of the cell is produced, ATP. So all these compartments are surrounded by one or two membranes. So the nucleus, for instance, is surrounded by two membranes. The mitochondria also are surrounded by two membranes, and the rest is usually one membrane. So if we zoom in, this is the plasma membrane here. What we have is, you can see that's a very complex cartoon. What we have here, this is the lipid bilayer that Marcus was describing this morning. So this is the exterior of the cell. This is the interior of the cell. And what we have here is the plasma membrane, which is made of lipids, and I will go to this afterward. And we can see many proteins which are inserted into this membrane. So these proteins, they are able to connect, for instance, the exterior of the cell to the interior of the cell. And some are very important because they are able to connect the cell to this exterior matrix, which is made of a lot of fibers, to another type of filament, which is called actin filament inside the cell. So it's like kind of a mechanical connector between the exterior and the interior. Some of them are also very important because they allow the cell to transport ions or molecules which are outside to the inside, because the membrane is also impermeable to big molecules. So some of them are like this molecule here, are like transporters. And And in addition, so we have the cytoskeleton. So the actin cytoskeleton is also connected to some other proteins via certain proteins to the membrane. And we have the macrotubule. So you can see that's a very complex system, very heterogeneous system, because it's made not only of lipid, but plenty of proteins. And it's something which separates the exterior from the interior. So it's a very important material, because it has to be mechanically resistant enough to sustain all the changes of shape that I described at the beginning. But at the same time, it has also many functions, because it has to connect mechanically the interior to the exterior, or in terms of transfer. So chemically, it has also be able to transfer molecules from inside to outside, or vice versa. So that's now I go back to more simple things. So Marcus explained this morning that amphiphilic molecule, it could be surfactant or it could be lipids. They are usually made of two parts. One, what is hydrophilic, which likes water, and the hydrophobic part, which doesn't like water and prefer to be sheltered, to be protected from water. And as he explained, there is a great advantage to self-assemble when you have this kind of molecule, and to form, for instance, bilayer when they have the right shape. And when you have a bilayer, so you can see here, this is a structure where you have the head group here, which are in contact with water, and the hydrophic chain, which are inside the bilayer. And a dimension which I think you have to keep in mind, that the thickness of a bilayer is of the order of 5 nanometers. So it means that this very thin layer, which is protecting the cell protecting the inside of the cell from the exterior, is only 5 nanometers thick, which is not much, right? And in addition, is existing simply because self-assembling rules, right? Just because the hydrophobic chain wants to be sheltered, to be protected from water. So if I want to go now to more detail about this lipid, so we can be a little bit more precise. So chemically speaking, most of them in cells are made of contain two chains and a head group, of course. And the chains could be totally saturated, or it can contain some unsaturations, as you can see here. And of course, they can also mix saturated and unsaturated chains. What is very striking is that if you look at the composition of cell membrane, they have a kind of a variety. You can see that the saturated chains have a kind of a number of carbon, which is ranging between 12 up to 20. And the unsaturated chains, I mean, can be diverse. They can have one unsaturation up to 2, 3 unsaturation. And I'm sure you have already heard, yes?
You mentioned that the membrane was strong enough to prevent the cell from undergoing pH changes. How much of that strength is coming from the membrane versus the actin?
That's a good point. So the question is, I explained that the cell can change shape a lot, and the question is how much comes from the membrane and how much comes from actin. A lot of this kind of deformation depend on cytoskeleton. And the beauty, I mean, what is very striking, I will show you after just in a few minutes that in fact the membrane itself doesn't sustain much stretching. So of course, all the deformation and come from active process like actin polymerization or sometimes other proteins as well. But the problem is the membrane has to sustain all these deformations, right? And that's the origin is cytoskeleton, but still it's mechanically to sustain all that. And you will see in a few minutes that's very surprising, because in particular you will see the membrane is very fragile in terms of mechanical stretching. Yeah, so I mean, I was saying that you can have a lot of diversity in a chain, and I'm sure you heard about omega-3 lipids. This is lipids which contain, which are very long chain and contain 3, 4, a lot of unsaturation on the chain. So basically, like here, many of them. So there is a large diversity, and so that's for the chains. But you can also have a large diversity in terms of head group. So typically, the lipids are categorized in three types. So those which are called phosphoglycerides. And the phosphoglycerides, the most common one is a phosphatidylcholine. I think we saw this morning a scheme from Marcus. So this is the most common lipid with this head group here. This is the chain here and the head group here. So this one is the most common. And you can have some of the head group which could have a smaller size. So just to come back to the shape that Marcus was introducing this morning, you can see that in this case, the head group is much smaller than this one. So this one will have a smaller shape parameter as Marcus was introducing. So that's one type. And you can have also lipids which are negatively charged, like the phosphatidylserine or this kind of even more complex lipids. But in vivo, there is no positively charged lipids. All the lipids are either neutral or sweet ionic or negatively charged. They can contain different charge. I mean, especially this kind of lipids can be transformed in with lipid with a more charge with special enzyme, but you never have natural lipids which have a positive charge. So that's the typical type of head group that you can have, and with a different mixing of chain, as I explained. And you can have also this lipid. So you can see here that the connection between the chains and the head group is different from here. And these lipids are called sphingolipids. They could have a head group which is the same as this one. It's called sphingomyelin in this case. And you have a large category of lipids which have contain a lot of sugar head group. And they are very important for signalization. These lipids here are very important. These lipids here are very important for signalization. Yes. That's a good point. I mean, I probably depends. I think that the design is such that these negatively charged lipids are used for the interaction with cytosolic protein on one side or the other side, so exterior or interior. So and usually the protein interact a lot through positively charged positive charge. So I think that that's designed that way, but and the only you can produce is very easy. That chemically prepare lipids which are positively charged, and people doing biotechnology, they are able to design this kind of positive charged lipid. But there is, even in bacteria, even in the ancient species, I think nobody has seen any lipid which is positively charged. That's a fact, and probably because the chemistry around is built around the negatively charged lipids. Yes, so I think that so these lipids are very important for signaling. They are used also for pathogens to attach to the cell. So they are able to target this kind of lipid to attach to the cell and to infect cells. And these ones are very important inside the cell. They are usually on the cytosolic part of the membrane, and they are used for a lot of active processes in cells, right? The OPC has double bonds here. Yeah. As one double bond on each chain. Yeah. So I mean, I don't want to spend too much time on this kind of, but you're right, that two double bonds. Yeah, and a very important one. So you can see in this case we have clearly this group which is usually ionic or charged, which is soluble. This chain which are insoluble. But a very important molecule, and I'm sure that Marcus will spend a third talk discussing a lot about cholesterol in membrane. And this is a very, you can see these interesting molecules, which has a very tiny OH group, which make it a little bit hydrophilic. But you can see also it has a very characteristic cycles here, which give it a shape here, which is very special which is very special, and give him properties for its localization inside the bilayer. And I'm sure that Marcos will detail much more that at least the importance of the cholesterol in the organization of the lipid in the membrane and the asymmetry of the membrane. So that's a very key parameter of cells. And the cholesterol is particularly important in the plasma membrane, so the membrane which surrounds the cell. And the cholesterol is not the same. You don't have the same composition in cholesterol in all your membranes. For instance, at the ER, there is no cholesterol, and there is kind of a gradient of cholesterol content if you go from the ER to the Golgi, which is a little bit more. And in the plasma membrane, you have typically like 30% cholesterol in the plasma membrane. But the localization of the cholesterol into the bilayer is still, a debate. And I'm sure that Marcus will have plenty of argument about that. So at the end of the day, so we have, and what I didn't mention also that you have more than 10,000 lipids which have been identified in cells. And so you don't have the same lipid in bacteria, you don't have the same lipid in yeast, etc. But altogether, even if you take a eukaryotic cell, you can have easily 10,000 type of lipids. And for the moment, nobody knows why you have so many of them, why you have such a diversity of chains, a group, etc. So at the end of the day, you have a combinatorial, which is very rich, but the reason why is still not clear. So at the end of the day, that's maybe a simpler cartoon of the plasma membrane, where you have, again, you have the membrane here with the protein which are embedded, the cytoskeleton which is underneath. And something I want to emphasize here, that the membranes are very peculiar material. Because they are, as I mentioned, it's a material which is very thin, typically 5 nanometer, but in this dimension is a fluid. It's like a two-dimensional liquid. So meaning that if now you are able to put a tracer on one of these molecules, it could be on a lipid or on a protein. So it means that you can see diffusing, and this has been done a lot by. It's been done a lot using different ways. So here, that's what you can see here. There's the case where quantum dots have been added to a protein. In this case, in fact, it was a protein. But so it means that you have a way to probe that the membrane is not a purely, let's say, frozen material, but the fluid system simply by tracking the position of lipid of protein in the cell. And if you do that, so you can see here the different technique which has been used to look at the diffusion of material, it could be proteins or lipids, into a bilayer. So you can add a small dye. You can add these quantum dots. So quantum dots are very small particles, typically 10 nanometer, which are intrinsically fluorescent. But you could use also gold nanoparticle. You can find different ways to label a molecule, and then you can look, as you can see here, you can track the trajectory. And this kind of technique has been used a lot to study the organization of the plasma membrane. People have a hard time to directly look at how the lipids are organized or the protein is distributed. And what they do, they indirectly label, for instance, lipid, and they look at the diffusion of the lipids into the bilayer. And doing that, you can see that you have cases where you can see here, for instance, a molecule which doesn't move, but you can see there are cases where you see long trajectories over time. And this tells you that simply looking at this kind of image here, that the organization of the plasma membrane is not homogeneous and it's very heterogeneous. Is okay? Yes. Yes. It's 10 nanometers, yeah. This case very simple with a microscope, a simple transmission microscope and fluorescence. That's simple. But you just need to have a sensitive camera to detect the fluorescence of this quantum dot. But there is nothing so difficult. So, I mean, I was cheating a little bit because what I showed you is something from my group, and it was not on a plasma membrane, it was on very simple bilayer, but it's exactly the same thing which has been done here in this kind of experiments. In this case, with at least it was, I think it was probably antibodies, fluorescent antibodies.
So, like, heterogeneity, if you place a tracker in the same exact position where it was like standing still in 10 hours, would it still stand still, or would it?
No, it will be totally different. So the cell is a system which is constantly remodeling itself. There is a lot of, I mean, as I said, there is a lot of traffic between the interior of the cell, which is bringing proteins and lipids to the plasma membrane. So, I mean, nothing is fixed in the plasma membrane. So if you take an image now and you take an image 5 minutes later, you don't get the same distribution. There is, in case of, when the cell is adhering, for instance, on the surface, this can localize sometimes some molecules which are used for adhesion. So this molecule, over a period of, let's say, 1 or 2 minutes, they won't move. But again, if you wait enough, there will be some transformation. So everything is moving all the time. But although it's moving all the time, usually you have you clearly see that the proteins and the lipids are not distributed in a homogeneous way. To put it that way. So that was my, at least so this cartoon, in fact, is exactly telling you this kind of a message that at the plasma membrane you might have heterogeneities. And this heterogeneities could be a small lipid domain. So we heard that there was I don't want to go into this debate, but for many years, people have looked at liquid phase separation in membranes, and they wanted to see if we can have lipid domain into the plasma membrane. So, and they eventually find that the plasma membrane contain some lipid domain, which they call lipid raft, which have a different, I mean, which have a typical size of between, let's say, 10 nanometer up to, let's say, a few hundred nanometers in general. So meaning that the distribution of the lipids is not homogeneous. The distribution of the protein also is not homogeneous. You could have some protein clusters. And there are many reasons why protein can cluster. So that's also, it could be because they are binding to a ligand. It's because they spontaneously interact between each other. So there are many reasons for which you can have protein cluster. And in addition of all this heterogeneity in the membrane, in addition, you have attached to the plasma membrane, you have this cytoskeleton, this, as you can see here, and this cytoskeleton somehow prevents the free diffusion of proteins into the membrane. So it can also contribute to the formation of protein cluster or protein or lipid domain as well. So yes. Yes, so if it's a tiny raft, it will diffuse, but with a weaker diffusion. Yeah. So that's themselves, and they're also very dynamic. It's not a domain which have a given size for a given time. So it depends a lot on the interaction of the protein which are in the raft with something else. So, but they diffuse as well. If they are big enough, very slowly. Probably. I mean, I don't know if people have looked at the dynamics of raft, but at least there is many good reasons why they can change shape, etc. So it's a little bit different from the condensate that we have seen this morning. But I mean, I could give a full talk about the raft, but I would say that for me, that's very interesting because the discussion that people have on condensate, 10 years ago, people have the same type of discussion about lipid domain into in plasma membrane, or at least in other cell membranes, right? So that's... Yeah. So maybe, yeah, we don't see on this cartoon, but there are plenty of protein which are able to bind the membrane and the actin. So some of them are kind of permanent bonds. Some of them you can have some chemical transformation which change the attachment of the protein to the actin. So you can have something which is transient. You can have permanent or transient bond between the plasma membrane and the actin filament. And they are themselves also very dynamic. The filament themselves are very dynamic. So it's why if you use the tracking of proteins, for instance, to explore the organization of the membrane, is very difficult because themselves they are exploring an environment which is heterogeneous, and in addition, the cortex underneath is also remodeling all the time. So it's why it's so difficult to interpret the diffusion data into the plasma membrane. I know. So that's so I didn't talk much about asymmetry, but so there is a lot of turnover because you have a lot of this vesicle I presented at the beginning. So you get a lot of them which are fusing, some of them which are internalized as well. So there is a lot of let's say influx and outflux of lipids into the bilayer, and it's the same for the proteins. And so you have a kind of steady state, I would say, which depends on all these fluxes, which depend a lot on the, I don't know what the cell is doing, the stage of the cell, for instance. So you get a lot of turnover, for sure. But at the end of the day, for a given, let's say, state of the cell, you have a steady state for the composition in the organization. But I mean, people say that the cell is able to renew its plasma membrane in an hour or something like that, in terms of flux of lipids. So it means that you need also a lot of production of lipids all the time, right? Yeah. So it was a kind of a brief introduction, but of course you could see from the first movie that one of the very key questions is to understand how is the mechanics of these membranes? Because if the cell has to be deformed all the time, to swell, to shrink, so what is the mechanics of membrane? And that's something which has, let's say, has been a very key question for the biophysicists since the 70s. So there are, I mean, that's there are some people. I mean, unfortunately, these two are. No, this one is still alive. Eric Zachman died not so long ago, and Evans is still there. So they wanted to understand how the mechanics of the cell was working, and at the time they wanted to work with to address this question with a simple system that the cell I've showed you at the beginning. This kind of a karyotic cell. So they wanted to have a system where they could just do some mechanics with at least the simplest as possible. And for that, what they had originally in the 70s, they were using red blood cells. So red blood cells, I mean, that's interesting because it's a cell which doesn't have any nucleus. It's a cell which is like a bag. For people, it's like a bag which contains hemoglobin, but still is a bag. It's a bag, but a bag with a given shape, and it has a very characteristic shape, as you can see here, with this kind of invagination. But it's not that simple because if you can see here, this is a plasma membrane of this cell here. You can see that you have, of course, not only lipid, but you have plenty of protein. And you have a sort of cytoskeleton, which is under the plasma membrane, which is, in this case, made of spectrin, not made of actin, but doesn't matter. So it's not that simple if you really want to understand simply the mechanics of the lipid bilayers, right? So, so they were developing tools to study mechanics, but they thought it was still difficult to address what was the intrinsic mechanical property of lipid bilayers. And fortunately, in the 80s, it was possible to prepare what we call giant liposomes, so which contain, for instance, one single lipid type, and then you could look at the mechanics. So what we call liposomes, that's a simple bilayer, so it's not a purely flat bilayer as Marcus was mentioning this morning, but it's a single bilayer, but forming a spherical structure. And I'm sure you already heard about liposomes for the creams, for instance. They use a lot of this kind of system. So basically, that's a material which is made of a single bilayer, which contain buffer inside, buffer outside. You can have what you want, and you can prepare liposome, which are called giant vesicles, but the point is that they have a diameter which is above a few microns. So typically 10 to 20 microns. So it means that there are objects which are now similar size as cells, and we can use some tools which have been developed for cells for the manipulation and the observation of these vesicles. And this kind of vesicle, is possible to prepare it with one lipid for one type of lipid, and you can control the type of lipid that you are going to put. So you have a way by controlling the material, the composition of a material to see how the mechanics of the object will be depending on the composition. You can also add a membrane protein. You can add different type of lipids. You can control a lot of things with this kind of object, and you can look. With that, you can look at the mechanics. You can look at many questions that you could find in inspired by biology. So, just to explain, so since they are typically 10 micron. That's good for microscopy, but the problem is in microscopy, usually if you look in the cell, you have a material, in particular the cytosol, which have an index of refraction, which is different from water. So you see them with a simple microscope. The problem with this vesicle, they have a tiny bilayer, 5 nanometer. The index of the bilayer is not so different from water. And so basically it's not so easy to look at. So somehow you have to find ways to increase the contrast. And that's something maybe that Marcus will love, to see some fluctuations. So what you can see here, that's a single bilayer. It's not a cell, a single bilayer, one of that giant vesicle, using a typical type of microscopy, which is called phase contrast. And you can see the contour of the vesicle here, and you can see the vesicle is jiggling. It's jiggling because of thermal fluctuations, right? And I'm sure you are going to describe that. Marcus, are you going to describe that on your? Yeah, so I leave it to him. But basically, it tells you that if you have a simple object like that under the microscope, so there is nothing living, right? Simple, the thermal fluctuation are able to deform the membrane enough that you can optically detect these deformations. So it shows you just visually that the membrane is a kind of a very soft material, because it can be active, it can be deformed by simply the fluctuation of the water molecules. Okay, so you could look at this vesicle with a simple microscope. You can do even better. You can add fluorescent lipids. So in this case, you can use confocal microscopy, and you can then do a lot of things with fluorescent images. Okay, so that's what we have in hand. Is clear? No question? Okay, so now we wanted to study with this kind of material the mechanics. So you can imagine that if you are interested in the mechanics of an object, there are three things that you would like to do. One would be to stretch it, something that you would like to stretch it, something you would like to bend it, and also something you would like to do is to shear. These are 3 types of deformation you could expect to observe in a material. So since it's the membrane, that is, these membranes are fluid, shearing a liquid is not very interesting, but at least you are left by measuring the stretching. And the question is how you can study the stretching of these vesicles. So maybe I will start with cells. So I'm sure you know that maybe you don't know, but if you take a cell and you put it in water, it will explode. That's clear? So, and this is exactly what people were doing originally when they wanted to study the stretching of the membrane. So when they were using red blood cell, they start from that. And there are 2 ways. Either you have an hypotonic medium, so meaning that you have a medium outside which is less concentrated than the medium inside the cell, or conversely, if now you put it in the more concentrated medium, the red blood cell can deform and have this kind of crenated shape. So that's one way to deform, to stretch the cell. In this case, you stretch it and you reach the point where the membrane explodes. And in this case, it's the opposite. You release the stretching and you can see that you change the shape. So, and why is this happening? This happens with cells or with vesicles because these membranes are semi-permeable component. So it means that they are not able to let ions flow or big molecules, so like a sucrose or whatever, or polymer. It cannot cross the membrane, but water can go through. So it means that if you have, so imagine that's your bilayer. So it can let water go through, but not the rest of the molecule. And if you have an unbalance in concentration, what will happen? You will get, in this case, you have let's say, you can imagine the interior of your cell. You have you have now less concentrated medium outside. You will have a flow of water which is going through the membrane in order to balance the osmotic pressure difference, right? That's and in this case, the difference of osmotic pressure is proportional, as you can see, to the difference of concentration that you have between here and here, with this RT coefficient. So that's what happens if you have a cell. Is what happens if you have vesicles. So you can imagine that it would be one way to stretch the membrane and to see how they behave when you stretch them. So you can do that with a vesicle, like the cell. So you can start from there, and you have, excuse me, so it will be exactly the same thing. So if we now have a system which, for instance, here, imagine that you have some sugar molecule, and you have a concentration which is lower outside. You have a flow of water, of course, so then you get the vesicle which will also burst. And if you do the opposite, you can see here that if you do this in a very fast way, often the membrane, or at least this kind of vesicle, are unstable. So if you make this kind of a very big shock, this one will be totally devastating because there will be no vesicle left. But if you put your vesicle and you do it in a very not in a very, you make it too rapidly, you can see that your vesicle, you can see here that the vesicle is not very stable. And so it's not a very convenient way to change, to stretch membrane or to play with the stretching, right? So something I wanted also to, I wanted to also to introduce this parameter here. So if you have your vesicle, which is fluctuating as we could see in the movie, so you can in fact define two parameters. One which is a real area of the membrane, which you can see this will be the real parameter, the real contour of the vesicles in this case, and you can also define what is the projected area of your membrane, and the difference between the real contour of the vesicle and the projected area, we call it excess area, and I'm sure that you are going to talk about that, Marcus. Probably no, maybe no, you're not. Anyway, but basically, it's possible to define a parameter which is the relative excess area as compared to the projected area of the membrane. And this is a kind of a parameter which tells you about, let's say, the floppiness, or at least the non-smoothness, I don't know if you say that, of the membrane. So practically, what we would like to do is to define a parameter which correspond to the variation of area of energy of the membrane when you change the area, and this parameter is called membrane tension, right? And this parameter, as you can see here, as a unit which is Newton per meter or joule per meter square, right? And if you take a piece of membrane and you start to stretch it, so you will change its area, of course, and you will, in this case, change its tension. And there is a regime so if you start stretching, there is a regime where now you have your membrane which is not which is not, let's say, ruffled anymore, but where you start to pull the lipid apart. And this regime where the tension now, the tension is changing linearly with the excess area. So this elastic regime is like a Hooke regime, right? And we can, with that, we can define the first modulus. So we heard this morning that Marcus was describing that the typical modulus that you can expect in life science. And so here we have a modulus which corresponds to this stretching, which is called a stretching modulus. And the question is, how is it possible to measure that with vesicles? Right? Yes. Yeah, but it's not, I mean, I just talk about I didn't describe here that regime, that transition here, right? So you are talking about that one. Yeah. Scale free or not? Let's mark. Yeah. I'm going to show you how we do that. So that's in this case, what you do, you need to externally apply some constraint onto the membrane, right? So it's not an internal. So it will depend, for instance, your vesicle, it will depend on the ratio between the surface and the volume of your vesicle. So when you, for instance, if you use this inflation-deflation technique with the water flow, you will change the volume with a given area, and then in this case, your tension will change, right? So it's not an intrinsic, it's not an intrinsic parameter of your bilayer. It will depend really on the precise condition of your system. Yeah? Okay, please. The difference between it's a surface tension, right? The difference here that if you think about surface tension about a droplet, so the surface tension, you can change the surface tension of a droplet by changing, for instance, if you have some molecules, imagine that you have oil droplets in water with surfactants, so you can define a surface tension, which depend in this case directly on the number of the surfactant molecule that you have, right? So so you can have a surface tension with if you have a surface tension, if you have a liquid and two liquid, you have a surface tension which is let's say in this case an intrinsic parameter of the two material. In this case, what we have is a surface tension, but where the value of the surface tension depends really on the detail between the area of your material and the volume which is inside. So it's not an intrinsic parameter. It's like not the interfacial tension between two liquids, which is an intrinsic parameter of the materials, the two liquids. In this case, that's a parameter which depends really on the system itself, how much it has been constrained. And as Marcus said, I will go after that to the bending modulus, which is a parameter which is also intrinsic of the material. But in this case, it means that if we have vesicles, its tension is not fixed. It will depend on how many lipids you have, so on the area and on the volume. That's it. But maybe I was not so clear, but it's not so easy to change this tension by simply changing the volume. So there are another technique I'm going to present now, which is much more controlled to change, at least to stretch it, right? And maybe you have ideas about how you can do that. Imagine that you have a vesicle or like a bubble. For some people, a vesicle it's not a bubble at all, but imagine it's a bubble. If you want to measure how it resists, or at least what is the property of the system when you stretch it, how can you do? Is there any idea about that? Any idea practically how you could measure this stretching? No. Okay, I will give you which. This is what, in a sense, is what we were doing with the swelling, but that one is not very controlled. So there is a technique which was invented, in fact, by Evan Evans in the 80s, first with the red blood cell and after that with a vesicle, which is very efficient. So it's like, imagine that you have a bubble, you want to stretch a bubble, you aspirate or you blow on it, right? But it's exactly the same thing. What he did is using, you can see here, this is again a giant vesicle, one bilayer. And what you can see, it has a pipette. So this is typically 10 macrons, this is typically a few macrons, and you can aspirate, right? And if we look more practically how it looks like, so here it's a microscope, you have a pipette, and I mean, in the old time now you have a lot of techniques. You have people doing macrofluidics developed a lot of system where you can aspirate with a given pressure, etc. So that's very let's elaborate. But a very simple technique which works very well is to use the hydrostatic pressure to aspirate. So basically, what you do, you have your pipette which is connected to a water tank, and you just go down. So you drop down your water tank, and the more you go down, the more you aspirate. So it's a very simple system, but which works beautifully to aspirate vesicle. And so I think that's one of the questions that Marcus put in his in this possible. So basically, it's possible to relate the difference of pressure, so the difference pressure that you impose by moving your water tank down. So you aspirate, and you can see that there is a simple relationship between the tension of your membrane, so how much you stretch, depending how much you aspirate. And this depends on the geometrical parameter. So dP is the diameter of the pipette, and dV is the diameter of the vesicle. So you can see that the more you aspirate, a linear relationship, so the more you aspirate, the more you stress. That's good. And then the question is now how much you are able to stretch. You have to measure it. And it's also very simple because you imagine that the more you aspirate, so the more your tongue here will be, I mean, the longer the tongue, right? And that is easy because, I mean, with a microscope, you can directly measure this tongue, this length here. So, and you can see that this would be this length. And the excess area that you get when you stretch a membrane, when you aspirate, you can directly measure it from this relationship. And I think that Marcus proposed you to calculate that, I guess. No, not this one. No, not this one. Okay, but if you want, you can find it. It's not difficult. And you can see, again, this is a very simple relationship. So I think it's a very simple system where you can directly control how much excess area you get when you aspirate. And if you do that, so this is a typical experiment done by Evan Evans. So you can see that you can measure the variation of the tension. So in this elastic regime, where you start to kind of move the head group apart as a function of the excess area, and you can see that you have a nice linear relationship. And from that, you can measure the stretching modulus. So since anybody would usually, when is able to measure modulus, looks as though the modulus depend on the material. So we can play with the lipid composition. And so this is typically experiments which have been done by Evans. So you can see here that he measured this stretching modulus for different lipid composition. So here we have a short chain, 13 carbon. You can see that we have no chain even when we reach 18 carbon. And here is also probing the effect of unsaturation. And you can see that the modulus here doesn't change. So you have a chance also to see that the stretching modulus is, in this case, of the order of 250 millinewton per meter, right? So it's a very stiff material. And also, I mean, you can look different composition here. For instance, I was mentioning the sphingomyelin and the cholesterol. And you can see that if you go from, for instance, SOPC, which is here, and you add cholesterol, you see that you have a strong increase of the stretching modulus of the membrane, and even more if you have sphingomyelin. So this means that at least this stretch, yes?
It's a very stiff material. Is there some macroscopic material you can compare that to?
Yeah, yes, I think that, didn't put it, I think I remove it. I think compared to rubber, it's that's I think it's compared to the kind of polymer, this is also similar, right? That's I think I put it somewhere, but I think I removed it. But anyway, it tells you that yeah. So maybe we should look at the if you look at the scale here, we have something like 3%, right? So it's not much. So you see that you need to exert. I mean, if you look at the quantity is quite stiff, and I would say probably of the order of the rubber, something like that, right? Right. And you don't stretch much. So basically, it was a few percent, right? So and the point is now, what happens if you stretch too much? So that was a question that we had before. So if you stretch too much, so you can plot for different composition. You can see that you can measure this tension versus this excess area, and you can see that all for different composition we have this star, and this star means that the vesicle explode, right? So the vesicle breaks. So it means that, and if you look at the scale here, that we can what is possible to reach is of the order of few percent, 4-5%, and after that the material break. It's because all these membrane, they exist simply because of self-organization. There is no covalent bond, which would probably increase their stability. But in this case, they exist only because of the self-organization, which results of the amphipathic nature of the lipids. And when you stretch too much, what happens is you create a hole, and the hole eventually lets water grow. And if you don't relax the material, the vesicle is broken. So it means that if I go back to the question that we had about actin at the beginning, a few minutes ago, it tells you that it's very frightening because you have a material which is very fragile, which cannot sustain more than a few percent of stretching, and it's stable only because you have a cytoskeleton underneath, and the area of the lipids, the bilayer, is much larger than the surface of the cytoskeleton. So you can imagine that it's like a system where you have a shell underneath and a membrane which has a lot of excess area. And when you stretch your cell, when the cell is migrating or the cell is dividing, so what happens is you first stretch the actin cytoskeleton, and you don't stretch the membrane. And there is a lot of system to protect the cell. So first, the excess area. You have some excess, let's say, some reservoir of membrane also, which in a membrane, which are there to buffer this kind of excess stretching. And that there is a lot of protection because it's a very fragile material. Okay, so that's so at least it has to be good for something. So what I tell you, that's not possible to stretch too much, but the question is it possible to deform it in the other direction? So is it possible to bend it? And so I think that's very nice because this morning Marcus ended his talk telling you that in fact it's possible to describe the mechanics of the membrane in a continuous way. And in particular, it's possible to describe how much energy is required to bend a membrane. And that's a very important expression which was proposed by Elfrich, where he proposed that the energy for bending a membrane depends on the square of the mean curvature of the membrane, and with a parameter again here, which is called the bending modulus. And this energy is per unit area. And so if for instance, if you take a cylinder, the mean curvature will be 1/r, the radius of the tube or the cylinder. And if you have a sphere, the mean curvature will be 2/r. So basically with that, you could in principle calculate how much energy, you can calculate and you can measure and interpret data when you bend the membrane. So first I will give you order of magnitude. So this bending modulus vary between a few kT for up to typically 100 kT, depending on lipid composition. And now, in this case, I put a comparison. So imagine that you are able to make a plastic bag, like a polyethylene bag, which will be 5 nanometer thick. The energy to bend this to bend the plastic bag will be 1,000 times stronger than what you need to bend a lipid bilayer. So it means it's easy, very easy to bend. And something also which I think could be interesting is I told you that in the cell you need to produce a lot of vesicles for trafficking. So how do you, and how much energy does it cost? So if you imagine that you have this expression, it's very easy to derive how much it costs to form a vesicle, right? So you just make the integral of this bending energy over a sphere. So you just do the very simple calculation, right, over a sphere. And what you find that the energy to form a vesicle is equal to 8 pi kappa. So it doesn't depend on the size of the vesicle. But, and if you can put numbers, so if we take a very relatively low bending rigidity, you can see that the energy to form a vesicle is on 250 kT. So it means that this is not something which can be possible simply by based on the spontaneous deformation of membrane, and you need proteins to do that, right? It tells you that if you want to form a vesicle, the membrane itself cannot do it alone, it just needs proteins. And if now I look at the typical, how the bending modulus depend on the composition of the membrane. So you can see here, this is a different modulus which have been measured depending on the different compositions. So this is composition where the chain length is increasing. So you can see in this case, so now the longer the chains, the more difficult it is to bend the membrane. And in addition, now if we change you have a chain of 18 carbon and you change the number of insaturation, you see the more insaturation you have, the easier it is to bend the membrane, because it's like you create some defect into the bilayer, it's easier to bend. And in addition, also if you add cholesterol, the bending rigidity increases, and similar if you have a sphingolipids. So that's so it's completely different from the stretching modulus, right? Yes.
Images, right? Either by looking at the curvatures or at the placement, right?
Maybe I will have time to show you how. Yes, I will have time. Yeah, I will show you. There are different techniques. So the micropipette. I'm not going to talk about micropipette, but this is, I mean, Evans, who was measuring that, was using the micropipette technique, but he was using the regime, and I didn't want to describe that today. But the regime where the membrane is more fluctuating. So he was using the pipette experiment to deduce his bending rigidity, and I will show you another way to do it. But you can experimentally measure it. So I will just show it afterward. It's not difficult. I would say the pipette experiments is not complicated. It's not a PhD life. It's more like maybe for one composition, it might be 3 weeks. Okay. And with the technique I'm going to describe later, that's not too complicated too. That's what is very dear. I mean, in fact, in micropipette experiments, the bending rigidity is easy to get. What is not easy to get is the stretching modulus. Because the problem of the stretching modulus is if you want to measure this properly, you need to have a membrane which has, so you have to be sure that your membrane doesn't have any, that you probe only very often when you prepare vesicles, they are not very smooth. They have a lot of defects. And when you start to stretch, you at some point you stretch the defects themselves, which become gradually flat, right? So I mean, you can have intrinsic defects in your membrane which are not part of the material. You see what I mean? If you have something which is not, if you have originally some tubes or something like that in your membrane, and you start to stretch it, there is a time where you are going to unfold these tubes, and this somehow perturbs the membrane and the measurement. So in fact, it's more complicated to measure the stretching than the bending modulus. Okay? Well, I will go to another technique. Anyway. Okay, so I told you that it's not easy to stretch, and we can it's possible to measure the bending modulus, and something which is probably the most striking example is to show that if now, instead of stretching, you start to pull, you will see that the vesicle is not an elastic balloon, but is something else. So imagine that you have your vesicle and you apply local force. So what you will get is it's a cartoon that I will show you in a movie in a minute. So what you are going to do is instead of having the simple vesicle, you are going to form a tiny cylinder, right? I will show you in a minute how it does. So so the experiment is the following. So we have a vesicle. Maybe I should stop that. So we have a vesicle. Like before, we have a macropipette. But now the difference is that we have a bead, and the bead is able to bind to the membrane. So it's able to adhere. And the bead is trapped by an optical tweezer. So the bead can move, but it's trapped. And we are going to move the pipette away. So if you do that with let's say an elastic balloon, what happens is you will deform the vesicle, and then it will just break, right, at some point. And now, because you have a fluid membrane which can be deformed in the other direction, what we will have is so on the left we have let's say this is a phase contrast image. So we see the pipette, but it's difficult to see the tube. In this case, we have it's another movie, and you see the tube here. So I will try to restart that. So if you look here, we have the contact between the bead and the vesicle. And now we are going to move the pipette away. And you see the deformation here. And you see that you have a tube which is formed. So it means that, even though it cannot support more than 5% deformation in the plane, in this direction is able to deform a lot, and which is not possible if you don't have a fluid membrane. Yeah. Question? No? Okay. And so just a typical dimension that we have. So I remind you, here we have 10 macrons. Here we have 4 nanometers. And what is very nice in this system is that this tube can be as small as, it can have a radius of 7 nanometers. So it means that the membrane can be bent to an extent which is very close to its own thickness, right? So it's a lot. And the tube also can be, depending, we'll show you next. It can also be as big as a few hundred nanometers. And how can we change the diameter? So, Marcus, this is, I know, so you have to calculate that if you want. It's not difficult. From the bending energy of the membrane, you can calculate the radius and the force which is required. And you can see that the radius of this tube results, in fact, it's a compromise between the bending energy, which is required to form the tube, and the tension of the membrane, right? So you can see that it's a result, and the tube is a ratio between kappa and sigma. And you can see that it's very easy to change the radius, because if you aspirate the vesicle, you will increase tension. And if you increase the tension, you will just change the tube radius. So that's a very easy way. So it's a system, at least that's convenient for that. And so you see the force also depends on these 2 parameters, the bending rigidity and the tension. So you can see that the higher the tension, the higher the force. But you can see that if you measure the force, because we have an optical tweezer, with an optical tweezer you can measure force, you can see that's a direct way to measure bending rigidity, which is not so difficult. And I will show you how we do. The length is a radius.
The radius of the big...
of that.
Oh, the radius of that.
Yeah. So I think I put it here because meaning that we can control that with the aspiration. But this is a radius of that. This one, this by preparation, we don't control anything. But that one, we control. And you can see that now that we can have a way also, if we measure the force as a function of tension, we can measure the bending rigidity. So this has been calculated directly, theoretically, excuse me. And this is what different people, like it was Jacques Pro and Ray Goldstein at the time, where they calculated the variation of the force as a function of the extension. So when you move the pipette away, so you first deform your membrane, and you can see that the force is increasing linearly. And you can see here maybe I will show you, some images. So basically here, what we start to deform to move the pipettes away. So we start deforming the membrane. So you have an elastic regime where you increase the force, and you see you have an overshoot of the force, and then at that stage you have a transition where you form the tube. And you see when the tube is formed, when you move you extend your tube, the force remains constant, because the force depends only, as long as the tension is controlled, you can see the force depends only on the kappa and sigma, but doesn't depend on the length of the tube, right? This is a profile also which was calculated with the same type of model. So we were trying to do, it's possible to test that experimentally. So this is typical experiments where you measure the force as a function of the displacements of your pipette, and you can see that we have exactly this overshoot. So this is when we extend the tube, and this is what we have when you retract the tube. And you can see we have exactly that, the force as an overshoot. So in our case, the overshoot is bigger than in theory because of this depends on the area of contact between the membrane and the bead. So that gives you the height of the overshoot. But when we form the tube, you can see that now you have a tube which is extended, so the force remains constant. And you see that that's totally, there is no hysteresis except here, because now is when it depends on the contact between the bead and. So that's totally in agreement with the theory. And from that, of course, you can measure the bending rigidity. And you can see that if you plot the force versus the square root, or the square of the force as a function of tension, you have a nice linear variation, and you can measure bending rigidity for this. It's a mixture of lipid that you can. Yes. So you calibrate. So you have the optical trap, and the optical trap you can calibrate, and then you can directly measure the force. Yes. No, yeah, the overshoot depends on the surface of contact between the bead and the vesicle. So the higher this surface of contact, the higher the overshoot. And in theory, they are the punctual force. So even if you have a punctual force, you have this overshoot. But you can see that the overshoot is typically 13% or something like that. That's a signature of the transition, right? Yes, on between that vesicle which is deformed and the tube formation. Yes. Don't change in this case. In this experiment, by aspiration, we just change the aspiration. So the more we aspirate, the higher the tension. And then from that, you can directly and you monitor the force, the corresponding force, and then you get
Aspiration is the
sucking. Yeah, this is sucking. So and that is easy because you just lower the position of your tank, and you measure the height, and then from that you have the tension. Yes. Can you speak louder? Do they have what? I should go closer. Sorry. I think I've become stuck. Yeah, so understand. So I think that's So if we pull the tube, do we change the excess area? Actually, so what happened is the material which is pulled in the tube comes from the tongue in the pipette. So if you pull a tube which is long enough, so if you calculate the area of the tube is very tiny. So, but in principle, if you pull a tube long enough, you should see your tongue which will be retracting, right? And we have pulled a tube which were like 500 microns long. So as long we can hold, and as long we can control the tension with the pipette, the force is constant. Yeah. No, I think when the bead, no, bead is just to make, when you pull, you need to have a good adhesion between the bead and the membrane, right? So this contact is just to make a mechanical contact between the bead and the vesicle. Yeah, but the bead remains, the attachment remains the same, doesn't change. But at some point, so you can imagine that you have an area of contact. So now to create the tube, you have to form the membrane over, I mean, at least the propagation of this deformation has to occur over a longer distance, right? So you have to deform, to pull more to get a deformation which is compatible with the tube. So that maybe I'm not clear. Yeah. Yeah. Yes, it's always in contact with the membrane. If you lose the contact, the tube is immediately retracting. It exists only because you have the contact between the bead and the membrane. If at some point, for instance, your experiment doesn't work well, so your adhesion is not strong enough, so they will detach and the tube will retracting. Yeah. But the linear regime before the overshoot, this corresponds to the deformation, to the deformation of the vesicle. There's an elastic deformation this one. Yeah.
No tube in that.
Yeah. Exactly. And with let's say an excess of the extent of deformation will depend only on the area of contact. That's it. Maybe I will start now to explain what happened with proteins, right? So I told you that it's possible to deform membrane with an optical tweezers, with changing the osmotic pressure, etc. But I mean, in real life, that's not how it takes place. So of course, we need proteins to deform membrane, and it's what at least what was exciting us for. So maybe I wanted to introduce again this kind of processes which happen at the plasma membrane, which allows the cells to internalize, let's say, component or internalize protein, which are the plasma membrane. And this phenomenon is called endocytosis. And so it consists of forming a vesicle which will be eventually internalized into the cells. It could be a vesicle, it could be a tube. And you have a process which depend I will show you a system which is called clatrine, and some which are independent of clatrine. And so that process where you form a vesicle and where you use the assembly of a cage that you can call a cage, which is a clatrine that you can see here, very organized, which is called a clatrine coat. And you can see that you have different steps where this vesicle is formed, where the cage is assembled. And you can see also with EM here. Gradually, we have a membrane which is forming a vesicle, and at some point, which is detaching. So that's something which depends on the assembling of this two-dimensional structure, very nice, which is called a clatrine coat. And there are cases where it doesn't depend on clatrine. And you can see different examples here where you have, in this case, oh, it doesn't work. So you can see cases where you have some protein which are internalized, which are toxins, or some receptor which are internalized. And that's too bad, it doesn't work, this one. But you can see that you have formation of tubes, in this case, not vesicles. And I wanted to show also that you can have also tubules in mass. Okay, but anyway, during trafficking, you can have tubules formed not only by an optical tweezers, but you can see here you can have naturally a vesicle tube which are formed. I didn't didn't work. Okay. But anyway, so it means that in vivo, you don't have any optical tweezer, but you can have tubules formed and vesicles. So for that, I have to introduce the fact that proteins, when they bind to membranes, they can, in some cases, deform the membrane. And one way to visualize that is to take a small liposome. So now I'm talking about liposomes, which are typically 100, 200 nanometer diameter, and that are exposed to protein which are in the bulk. And these proteins can interact with the membrane different ways. So some of them can insert between the head group of the bilayer, and you can see what happens if you have them outside, and you have a typically macromolar of these proteins. You can see that you transform these spherical vesicles into tubules. That's true also, even though the proteins don't have shape, they sometimes have a structure. And if you have proteins which have a banana shape, as you can see here, these proteins. So again, if you incubate the liposome with these proteins, you can see that you transform the spherical vesicle into tubules. And you can see that if the protein have a banana shape, which is not too much curved, you have a wide liposome, wide tubes. And if the protein is more curved, you have more tiny tubes. And it's not only for soluble protein, but if you have proteins which are embedded in the membrane, which is called transmembrane protein, but that have a conical shape, and this is a case for this kind of protein here, you can see that when we reconstitute the proteins into the liposome, they can also have a tubular shape. So it means that in the absence of force, it's possible to transform a membrane into a tubule. And to account for this kind of deformation, what we have to introduce is what we call the spontaneous curvature of the membrane. So the membrane now, if it's flat could be a zero curvature in presence of this protein an asymmetric way or that have an asymmetric shape, you can see that the membrane now is curved, and the curvature of this membrane is called the spontaneous curvature. So in principle, you can imagine that the more protein you have, the higher the spontaneous curvature, right? So you expect that this quantity, C0, will depend on the surface fraction of the proteins. And at least there is an interesting example, at least which has been treated properly. There's a case where you have these proteins which have a structure here, which is able to fold as a helix, and this helix is like let's say macrosurfactant. So it has one side which is hydrophilic, one part which is hydrophilic, and another part which is hydrophobic. So when this protein is folding on the membrane, it will introduce the hydrophobic residue into the bilayer and let the hydrophilic residue outside. So at the end of the day, it can insert into the head group. By inserting into the head group, it will occupy more surface on this leaflet than compared to this leaflet, and this will impose a bending to the membrane. So there's a calculation, which was a simulation calculation, which have been done by Felix Campello when he was working with Misha Kosloff, and he calculated how the spontaneous curvature depend on the surface fraction of inclusion that he had on the membrane. And you can see that there is a linear relationship between the spontaneous curvature C0 as a function of this quantity phi, that the surface fraction, up to typically 10%. So it means that the more you have, the more you bend the membrane. And you can see that here. That's, yes. So the color is more the stress into the bilayer, which are created by this insertion. So that locally it creates and interestingly, this stress will depend how deep the amphipatic helix is inserted into the bilayer. So they calculated how much deformation you get depending how deep you go. You can imagine that if you have this amphipatic helix in the middle, everything becomes symmetric and the deformation is zero, right? If it's in the middle. And the farther away you are from the middle, the more deformation you create. It's exactly what happens. So what they were calculating is a deformation, the spontaneous curvature, as a function of the position of the amphipatic helix compared to the center of the bilayer. And you can see, of course, if the protein is outside, there is no deformation. And you can see there is a maximum when the helix is weakly inserted, and the deeper you go, the smaller the deformation. The finite side of the deformation of the bilayer? No, oh, you mean in this in the simulation? Probably, but I don't think the size is important. No.
Does it continue on treatment?
Ah, yes. I think so. I think the continue, but that in this case, it was more like maybe no, maybe I don't get the question. Okay, yeah. Insert, yeah. Maybe the spontaneous curvature again. It's a kind of continuous description, which I think is still valid, at least in the. I mean, even maybe it's misleading with this kind of a cartoon here because they were just drawing the deformation, but at least this shows that this model is still valid because I mean that's a continuous description. Maybe I'm not correct, but.
You're right. You will have to think very hard about
if it's, but if it's a tiny one, but I'm not sure they were. Maybe you.
You have to think about fix it like this.
Ah, yeah.
Then,
I know for sure that's for sure. Yeah, yeah. No, that's true. That's true. Yeah. It's not a solid; it's a fluid.
No, you're right, but like, so is it close to the usual shielding solution that you see in
Maybe I don't understand.
So you can imagine a solid.
Yes.
And you put
yes.
Solid is infinite. You can find analytic people.
Yes. Yeah.
So it's different.
Yeah. No, I think it's completely different. But maybe that you have something about the stress. No. Yeah.
Like solids, you have shear stresses that are non-negligible. You have to account for those. Those don't show up here.
Yes. You had a question about it? Or?
Yeah, I'm confused why it's C.
The
left.
On the left, this one?
Yeah, like I have a on top, I would have might misunderstood.
That position is here. So why is deforming here more than that? That's your question.
Or even in the compared to the bottom, like why is the bottom?
Is bending in the other direction. That also depends how you describe the coupling between the two bilayers as well. So I think that's. I mean, I don't want to be exact. It's just. I mean, they have also another model where they consider now that you have a coupling between the two monolayers, etc. So the point was to say that the depth of the inclusion will influence the spontaneous curvature. So it's not a parameter per se, that the spontaneous curvature, but will depend on that too, on the concentration of proteins.
Surface coverage by these or insertion that way.
Not so easy to do. Huh? So actually, the problem is that all these proteins, they don't have only an alphabetic helix. They also have some bulky part, and often you have another effect which is due to the what people call crowding effect. So you could have also some other effect when you concentrate them, so which also can influence. So I think that to do experiments with only alphabetic helix is not so easy. But in principle, you could do it, but it has not been done. What we have done is with bar domain. No, so it's what we did with bar domain, for instance, what people have done with bar domain, because it's much easier to control, but I think it will be for next time. So maybe I'll show you what happens, so what people have done, but that's to simply incubate again the vesicles with these proteins with amphipathic helix. And so if they can have only the part which contain the amphipathic helix or the full length, and you can see that you have tubules. But with that, the only thing you can do is measuring the radius of the tube. But at least in principle, you should work with different proteins to see if you see a difference in the tubules. But that was not done systematically. There was no systematic. Yeah. The bending. So that's a good question. So the bending. So the question is why the bending rigidity doesn't depend on the inclusion. This was your question, and you can describe. You can make a model where you kind of renormalize your bending rigidity by the inclusions or by the protein. So that you can also do a model that way. Is one way to describe the system. So there are and it's probably much more true if you have protein which are in the membrane. So in this case, it's in between because it's a protein which is weakly embedded. But for sure, when you have protein which are really part of the bilayer, for sure, you have to take the protein themselves into the bending rigidity. And in fact, I hope next time I can show you that's something which has been done properly. Yeah.
About how normally the membrane kind of has a solution, and then there's the bending or spontaneous bending associated with the inclusions. How does the size, the length scale of that compare with the
but the problem of the deformation, they exist at all scale. So that, I mean, I've shown you optically, you can distinguish long wavelengths deformation, but you have deformation at all scale. So that's and the curvature that you can have will depend directly on the type of protein you have. So I'll show you cases where the deformation are of the order of tens of nanometers. So the tubules, but you can have also cases where people have shown that you could have deformation which could be micron size. So that's so it depends on the type of molecule that you use. With filament, for instance, some filaments you can have micron size. Maybe I stop. Oh, I stop here. Yeah. This time. More question?